Okay, welcome to Unit 5, Heredity. I'm so excited because this is arguably my favorite unit. I think you guys are really going to love it. And we're going to start out by talking about Mendelian genetics. So just a little background, I'm not sure where you're listening from, but if you're one of my specific students, I know we've just come off of a winter break and we had covered some genetics topics before then. So this is going to be a little bit of a review, hopefully. So if you remember our gametes, which are egg in females and sperm in males, with the exception of those, all the other cells in your body are diploid. And remember, think diploid double, meaning they have 2 copies of every chromosome. 1 copy of each chromosome came from your biological mother and her egg. 1 copy came from your biological father and his sperm. Now remember, humans have 23 pairs of chromosomes. So 23 individual copies from mom, 23 individual from dad. Those 2 sets came together when they fertilized and made the first cell that was you, the zygote that had the 46 chromosomes that make you who you are. So 2 of each of the 23. So a couple other terms we need to review. When we're talking about chromosomes and DNA, we have to talk about genes. So remember, your DNA is organized into chromosomes, bundled up into chromosomes. And sections of those chromosomes or sections of your DNA are genes. A gene is a specific section of your DNA that is going to provide the instructions for making a polypeptide, which will fold into a protein. And since a gene is a piece of a chromosome and you have 2 of every chromosome, remember in those pairs, 1 from mom, 1 from dad, you have 2 copies of every set of instructions to make every protein in your body. So you have 2 copies that are working together to figure out what protein ends up being made. And so what we're going to talk about in this unit is, okay, how do those instructions work together to figure out which ones we follow and which ones we listen to? So here's a new term, alleles. An allele is a different version of the same gene. So for example, we inherited a full set of chromosomes with all of our genes on them, thousands of genes from each of our parents. So we have 2 sets working together. But what you inherited on chromosome 21 from your mom may not say the same thing as chromosome 21 from your dad. So for example, mom may have given you a gene for blue eyes and dad may have given you a gene for brown eyes. So we're looking at the gene of eye color, but the alleles could say blue and one could say brown. Now, eye colors is a little bit more complicated than that, but that's just an example of what we mean by alleles. And again, what we're going to learn about in this unit is how those different alleles from mom and dad, work together to make you who you are and the traits that we see. Another review, so when we're talking about these pairs, remember we're talking about homochromos or homologous chromosomes. So those are the matching or the coordinating chromosomes from mom and dad that have the same genes on them in the same locations, but the genes may have different alleles. They may say different things. Okay, so the more we talk about this, the more it's going to make sense, I promise. So that's just a little review. Let's dive in a little bit more. So the title of this concept is Mendelian genetics. So I would be remiss to not mention who Gregor Mendel is, since that's what this is all titled. So he was an Austrian monk. And what he did was he loved plants. And so he used pea plants specifically to study and learn about genetics. And he is known as the father of genetics because what he discovered about these pea plants set the foundation for what we know genetics to be today. And he basically organized his observations into 3 laws of inheritance. The law of dominance, the law of segregation, and the law of independent assortment. So we're going to go through each of these. And these still to this day are the foundation of what we know about genetics. But we've also learned that some traits are a lot more complicated. And so we'll learn those exceptions later on. But for today, we're just going to learn the basics. Think of today as like Heredity 101 or Inheritance 101. Okay, so what did he do? How did he figure this stuff out? He did a ton of experiments with pea plants. And when he crossed them, when he mated them, he made sure of a couple things. One, he made sure he had control over the breeding. He specifically used pea plants that typically self-pollinate. So so that he could make sure like he knew who was crossing with what. So it was all very, very controlled. Remember from Unit 1, we've talked about how important it is to have controlled variables in our experiments, and that's what he was doing. He also only used purebred plants, and that term in genetics is referring to organisms whose ancestors are genetically uniform. So for example, when I'm looking at this picture and he crossed a purple flower with a white flower, this would be a purebred purple, meaning all of its ancestors that he knew were purple, and these white, all of theirs were white. So we're looking for ones that way for him to study. And then last, he looked at either or traits, meaning you really only looked at simple traits that there was only two options. And there's a lot of things like that in pea plants. So for example, in peas, the shape of the peas can be, round versus wrinkled. That's what the pea color, the flower color, they can be purple or white. In general, the height of pea plants can be categorized as tall versus short. So he's looking at these either or traits. He was studying really simple things. We have either or traits in us as humans as well, and we'll look at some, but a lot of our traits are more complicated than that, which again, we'll get into in later concepts. So I mentioned that he was crossing the pea plants. What do I mean by that? A cross is just the mating of two organisms. So you start with your pea generation, that's your parental generation, and this is where Mendel was using purebred. So this was a purebred pure flower with a purebred white flower. And when he crossed that parental generation, he got the first generation, or the first filial generation, which would be like the kids. And he saw that they were all purple. He was like, what? I crossed purple and white. How did they all end up purple? Then he took this offspring generation, this F1, he crossed them with each other. So two kids together. And in his F2 generation, when he was doing this over and over again, he found out that 75% ended up being purple and around 25% ended up being white in the flower color. And so he did this hundreds of times. And this is the general probability that he kept saying over and over again. And this is the type of experiments he used to figure out what he did about genetics. So what did he figure out? Okay, well first, let's go back to that word alleles, the different versions of a gene. In order to refer to them, we have to assign letters to them. So we use uppercase letters for dominant, example, a capital A, and we use lowercase for recessive, so example, a lowercase a. So let's say we're looking at gene A. If I'm talking about a capital A, I'd be the dominant allele. If I'm talking about the lowercase A, it'd be the recessive allele. So what do I mean about dominant and recessive? We will get there, I promise. Remember, you're getting two alleles, one from each parent for every gene that you have. So let's say you get the same alleles from your parents. So you get two dominant alleles, or you get two recessive. So one from mom, one from dad, they're the same. We say that that is a homozygous. The zygote was homologous. They were the same. In your zygote, you got the same versions of the genes. Hetero means different. So heterozygous means your zygote got two different alleles. One parent gave you a dominant and one gave you a recessive. So notice the different colors I'm using to represent these genes to show heterozygous. Okay, so those are two critical terms. Now, let's talk about that dominant recessive. This is Mendel's first law, the law of dominance. And what it says, what he discovered, is that a dominant or a strong allele will express itself over a recessive weak allele. Okay, what do I mean? So dominant, if present, if you have a dominant allele, it's going to always be expressed or seen. That's why I refer to it as strong. Okay, so if you get one, it's going to show. So for example, if brown hair is dominant over blonde hair, which is true, hair color is a little bit more complicated, but in general, brown is dominant to blonde. Little b would represent the allele for blonde hair, for recessive. Capital B would represent brown hair, for dominant. If you get a big B, a dominant allele, from your mom or your dad, or from both of them, you're going to have brown hair. So whether you get two big Bs or just one, you're going to have brown hair, because in this combination where you got a brown allele and a blonde allele, brown is going to dominate. It's going to be stronger over the recessive. Recessive means the allele is going to only be seen or expressed when the dominant allele is not there to mask over it. So the only way to get blonde hair is to inherit that little b allele, that recessive allele from both parents, little b to show it. Because if you get the dominant, it's going to mask over it. That's what we mean by dominant and recessive. It doesn't mean more common versus less common or better versus worse, not at all. Recessive just means you're going to need to have 2 copies of it to show, and dominant, you only need 1. Now, I've been using words like seen and show. Here's what I mean by that. Your genotype is the actual alleles you inherited. It's the genes that you got. So, for example, genes that code for the gene that codes for flower color, you could get 2 dominant alleles, you could get a dominant and a recessive, or you could get 2 recessive alleles. Phenotype is the physical traits that you see. So, for example, do you have purple flowers if you're a pea plant, or do you have white flowers? So the genotype is determining what the phenotype is, and the phenotype is what we actually see. So the genotype is your genes, and it determines what proteins are made, and then those proteins make up what we actually see in the phenotype. Another one of the laws that Mendel came up with from his experiments was the law of segregation. So what this says is that when chromosomes separate in meiosis, each gamete, so eggs, if a female is doing this, or sperm in males, is going to only end up with 1 chromosome from each pair. So for example, look at this picture. This organism only has 4 chromosomes total. If this was a human, this would have 46 duplicated chromosomes. So when meiosis happens, remember, it makes gametes that are haploid with half the amount. So it goes from 4 to only 2. Notice only 1 of the blue ones and 1 of the orange from each of the original pairs. In a human, this would be 46 X's, and then each of these would only have 23. Now, how does that compute to genetics? So let's say a man has alleles for brown hair and blonde hair. He is heterozygous, big B, little b for hair color. Okay? He has 1 of each. The sperm cells that he makes for his children are either going to end up with the allele for brown hair or the allele for blonde hair. He's not going to pass on both. So each sperm only gets 1 of the alleles, and then if that sperm fertilizes with an egg, that's where the 2 come together to make the offspring. Now, a mutation can happen and a mistake happens in this. It's called non-disjunction. We'll talk about some of those examples in Concept 3. But in general, this is what is happening in the law of segregation. And then the last law Mendel came up with is the law of independent assortment. And it's exactly what it says. The assortment of chromosomes is independent for each trait. So for one trait, it doesn't affect the assortment of chromosomes for another trait. Okay, so for example, when the chromosomes line up in metaphase and then are separated in anaphase, when they're lining up, they're not like organizing in the sense that like all the dominant chromosomes with dominant genes come on this side and all the ones with recessive go on this side. No, it's totally random. This means that any combination of your maternal and paternal chromosomes can be passed on to your future offspring because they're lining up randomly. That's why a couple can have 5 kids and they all are so different from each other because the combination of chromosomes is totally random and completely independent of each other. And because of these things, we're able to use a tool called Punnett Squares to make predictions about what offspring may inherit and then what they may look like. So a Punnett square is a diagram that shows the probability of our inheriting certain genes and thus certain traits from parents. We have monohybrid crosses and we have dihybrid crosses. So a monohybrid cross is just looking at 2 organisms and we're looking at 1 trait. So in the Punnett square, we put the genotype of the parents on the outside, so the different alleles. And then on the inside is where we see the possible genotypes of the offspring. So for instance, look at these 2 parents. Their genotype for gene A is heterozygous. This parent has a dominant and a recessive, and this parent is a dominant and recessive. And so when I fill in the Punnett square, I take 1 from the side and 1 from the top. That's where the 2 big A's came from. And then right here, 1 from the top, 1 from the side. That's where big A, little a came from. Here, 1 from the side, 1 from the top, and then here, 1 from the side, 1 from the top. Now, when we write it, we always just put the big letter before the little if it's heterozygous. That's what we're seeing here. And what we have on the inside here is we have possible genotypes of our offspring. And so what we can see here is that 1 out of 4 of these parents' kids will be homozygous dominant, meaning have 2 of the same dominant allele. 1 out of 4 will be homozygous recessive in their genotype, so 2 recessive alleles. And then 2 out of 4, or 50%, are going to be heterozygous. They're going to get a dominant and a recessive allele in terms of their genotypes. And then we can also look at the phenotypic ratio as well. So I think the best way to understand this is to practice it. So we're going to do some practice problems in class together. I want you to try these. In these examples, we're going to be looking at pea plants and the trait of round versus wrinkled seed. So in pea plants, round is dominant to wrinkled. So I want you to use the letter R to represent this trait. So round would be a dominant or big R, and wrinkled would be a little r for recessive. Okay, and I want you to try each of these problems and work them out, and then we'll talk through those together. Okay, last thing I want to mention for my non-honors students, and then I'll go a little bit further with honors, are dihybrid crosses. So we're going to break this up in a class over several days so it isn't overwhelming. But we're going to do dihybrid crosses when we're finding the possible genotypes for offspring, when we want to look at two traits at the same time. So for example, what's the likelihood that you get brown hair and blue eyes from your parents, or blonde hair and blue eyes? So our first example we'll work together is we're going to use tomato plants that are tall and red. So we're going to look at what's the likelihood maybe of getting a plant that's tall and red. In tomatoes, tall is dominant to short and red is dominant to yellow. And so we'll do some crosses here. And I'm going to walk you through some steps. You're going to find the genotypes of the parents first. Then you're going to sort the alleles and figure out the different combinations. I like to do FOIL like we do in algebra. And then you'll write those on the outside, you'll fill them in, and then you'll determine a phenotypic ratio. And we're going to do some examples in class together on this. So I'm not going to show you the answers because I want you to try that yourself. Okay, this last thing I'm just wanting to cover with my honors students. So if you're not in the honors class, don't worry about this. But if you're in honors, I want you to know that we can use the rules of probability, throw it back to math class, to determine the possibility of getting a specific genotype, and thus also then a specific phenotype. So I find this especially helpful if you're looking at more than 2 genes at a time, because then your Punnett square would just be massive and it would not be doable. So Punnett square is useful for 1 or 2 at the same time, but any more than that, you're going to have to use probability. So here's what we do. You'll do a monohybrid cross, so Punnett square, for each individual gene. And you're going to determine the odds of getting each genotype in fraction form. And then we're going to figure out how common it is to get this and this and this will multiply. So and in math means to multiply to get in terms of probability. And so that's what we'll do to get the likelihood of getting all of these things together. So here's what I mean by this. So let's say Erica has this genotype for 3 different genes. She's homozygous dominant for gene A, heterozygous for gene B, and homozygous recessive for gene C. And Carlos has the genotype heterozygous for all 3 traits. We want to know the probability of Erica and Carlos having a kid who is heterozygous for all 3 genes. So what's the likelihood of getting a kid like this? Well, this Punnett square would be insane. So instead, we're going to follow those steps. We're going to cross them for gene A, cross them for gene B, and cross them for gene C, and then we'll work them all out together. So gene A, let's look. Erica's up here. She's homozygous dominant. Carlos is down here. He's heterozygous. Okay, so for gene A, there's a 1 out of 2 chance, or 2 out of 4, but we're just reducing it, that they are homozygous. Their kid is homozygous dominant, and 1 out of 2 chance that they're heterozygous. Gene B, they're both heterozygous for gene B. So that's the parents on the outside. Fill in the Punnett square. And let's look at the probability. 1 out of 4 of their kids would be homozygous dominant. 2 out of 4, which reduces to 1 out of 2, would be hetero. And then 1 out of 4 would be homozygous dominant. Then let's look at gene C. She's homozygous recessive. That's what's on top. He's hetero, so that's here. We fill it in. We get a 1 out of 2 will be homozygous, or excuse me, I'll correct this. It should say little c, and 1 out of 2 should be big C, little c. So then we're going to multiply. What's the odds of getting a big A, little a heterozygous? 1 out of 2. What's the odds of getting big B, little b? 1 out of 2, which we see here. And what's the odds of getting big C, little c? 1 out of 2. So multiply. 1 times 1 times 1 is 1. 2 times 2 is 4. Times 2 is 8. So they have a 1 out of 8 chances that they'll have a kid that's heterozygous for these 3 genes. That's how we can use probability, and we'll practice this in our honors course together. And that is our Intro to Mendelian Genetics.